The attempts to classify living organisms began centuries ago. The first classifications were made to identify organisms that were useful to man for food, shelter, and clothing. It was Aristotle who devised the first scientific method of classification. Based on morphological characters, he classified plants into trees, shrubs, and herbs, and animals into two groups, those that had red blood and those that didn't. Later, Carl Linnaeus came up with the two-kingdom system of classification: Kingdom Plantae, that included all the plants, and Kingdom Animalia, that included all the animals. This classification system was reasonable for that time, because it was easy to distinguish between immobile plants and mobile animals. However, this system did not distinguish between unicellular and multicellular organisms, prokaryotes and eukaryotes, and photosynthetic and non-photosynthetic organisms. There were also a large number of living organisms that did not fall into any category under this system. New studies about various organisms also revealed enormous variations of characteristics within the organisms of the same group, like cell structure, mode of nutrition, methods of reproduction, and evolutionary relationships. Therefore, though the two-kingdom classification system was used for a long time, it proved to be an inadequate system recently. In 1969, R.H. Whittaker proposed a five-kingdom classification system where three more kingdoms, Monera, Protista, and fungi, were added to the existing kingdoms of Plantae and Animalia. The main criteria that Whittaker used for this classification system were cell structure, thallus organization, mode of nutrition, methods of reproduction, and phylogenetic relationships. Phylogenetics is the study of evolutionary relatedness among various groups of organisms. The earlier classification systems included all bacteria, algae, fungi, mosses, ferns, gymnosperms, and angiosperms under the plants category, based on the common premise that each one had a cell wall in their cells. The five-kingdom classification remedied this by placing organisms in different kingdoms based on the differences in their characteristics. Based on the differences in cell wall composition, heterotrophic fungi differ from autotrophic plants and were grouped under Kingdom Fungi. All prokaryotic organisms were grouped together under Kingdom Monera, and unicellular eukaryotic organisms were placed in Kingdom Protista. Previously, unicellular Chlamydomonas and multicellular Spirogyra were placed together under algae in Kingdom Plantae. However, in the new classification system, Chlamydomonas moved to Kingdom Protista, while Spirogyra remained in Kingdom Plantae. The five-kingdom classification system not only classified organisms into different groups, but also put together organisms that were earlier placed in different kingdoms. For example, Kingdom Protista brought together both Chlamydomonas and Chlorella with other eukaryotes like amoeba and paramecium, which were initially placed in Kingdom Animalia. Therefore, the five-kingdom classification system was evolved with an advancement in our understanding of the characteristics of organisms and their phylogenetic relationships. Depending on more such discoveries, the classification system may further need to be evolved in future. Bacteria are the sole members of Kingdom Monera. Bacteria can be found anywhere, including extreme habitats like hot springs, deserts, and deep oceans. Different bacteria synthesize their food in different ways. Some of them are autotrophs. That is, they synthesize their own food from inorganic substrates. However, a majority of them are heterotrophs. That is, instead of synthesizing their own food, they depend on other organisms or dead organic matter for food. Based on their shape, bacteria can be classified into 4 types: the spherical coccus, the rod-shaped bacillus, the vibrio, which is in the shape of a comma, and the spiral spirillum. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. Cyanobacteria, commonly known as blue-green algae, are examples of eubacteria. They are unicellular, filamentous algae that live in colonies. Their colonies are usually surrounded by gelatinous sheaths. Bacteria can be divided into autotrophs as well as heterotrophs. Autotrophs can further classified into photosynthetic autotrophs and chemosynthetic autotrophs. Like plants, cyanobacteria have chlorophyll A, and they are, like them, photosynthetic autotrophs. That is, they synthesize their food by using light energy. Did you know cyanobacteria like nostoc and anabina can also fix atmospheric nitrogen in their specialized cells called heterocysts? Chemosynthetic autotrophs oxidize various inorganic substances such as nitrates or ammonia and use the energy released for ATP production. They help in recycling nutrients like nitrogen, sulfur, and phosphorus. On the other hand, heterotrophic bacteria are decomposers. They help us make curd, fix nitrogen in legumes, and produce antibiotics. They can also cause harm by damaging crops. And some diseases like typhoid, tetanus, and cholera are known to be caused by bacteria. Bacteria reproduce by fission. They can also reproduce sexually by a primitive DNA transfer from one bacterium to another through the pilus. Under unfavorable conditions, bacteria produce spores. Apart from eubacteria, there are special bacteria called archaebacteria that live in extreme conditions. For example, halophiles live in extreme salty areas. Thermoacidophiles live in hot springs, while methanogens live in marshy areas. Methanogens are also present in the gut of ruminant animals as cows and buffaloes. These bacteria are responsible for the production of methane, also referred to as biogas, from the dung of these cattle. All archaebacteria have a different cell wall structure than other bacteria, which helps them to survive in extreme conditions. Unlike eubacteria and archaebacteria, mycoplasmas are the bacteria that do not have a cell wall. They are also smallest living beings ever known who can survive without oxygen. They are pathogenic in animals and plants. Kingdom Monera, therefore, consists of eubacteria, archaebacteria, and mycoplasmas. All unicellular eukaryotes come under kingdom Protista. Members of the kingdom Protista are primarily aquatic. Chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans are few categories of protists. Since they are all eukaryotes, they contain a well-defined nucleus and other membranous organelles. Some protists also have either a flagellum or a coelom. They reproduce both asexually through cell fusion and sexually via zygote formation. Chrysophytes include diatoms and golden algae or desmids. They are microscopic and float in great numbers in both freshwater and saline water. Also, they are mostly photosynthetic. Diatoms are known as chief producers in the oceans. Diatom cell walls are embedded with silica, making them indestructible. Diatoms therefore manage to leave behind a large amount of their cell wall deposits in their habitats. These fossilized remains of diatoms form diatomaceous earth. It can be put to various uses like polishing and filtration of oils and syrups. Dinoflagellates are colorful protists found both in marine and freshwater. Depending on the main pigments in their cells, they are yellow, green, brown, blue, or red. Sometimes, when a red dinoflagellate multiplies rapidly in the sea, it makes the sea appear red. This red tide phenomenon is known to be harmful, as the toxins so released can kill other marine animals such as fish. Dinoflagellates usually have two flagella, longitudinal flagella and transverse flagella. Their names indicate how both the flagella are positioned. Euglenoids too, like dinoflagellates, have two flagella, one short and the other a long one. Instead of a cell wall, euglenoids have a protein-rich layer called pellicle that makes their bodies flexible. Euglenoids are phototrophic autotrophs in the presence of sunlight, since they also have chloroplasts, like plants. However, they behave as heterotrophs in the absence of sunlight. Slime molds are saprophytic protists, that is, they live on dead organic matter. They form a huge aggregation called plasmodium. However, during unfavorable conditions, this plasmodium differentiates and forms spores that possess true walls. These spores are so resistant that they can survive for years under adverse conditions. Protozoans are protists that are believed to be primitive relatives of animals. Did you know that the word protozoans means the first animals? All protozoans are heterotrophs and live as predators or parasites. They can be classified into four types: amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. Out of these, amoeboid protozoans are found in freshwater, marine water, as well as moist soils. They use false feet called pseudopodia to move and capture their prey. Some amoeboids, like entamoeba, are parasites. Flagellated protozoans, as the name suggests, have flagella. They can be either free-living or parasitic. Some parasites like trypanosoma can also cause diseases like sleeping sickness.